Good night. I'd like to make two announcements. First, I would like to thank FAPESP and PAL Corporation for the funding and financial support, respectively. And then I'd like to thank the affiliated societies for helping us to put together this scientific program of this meeting. I hope you are enjoying the program so far. Tonight, we're going to have the presence of Dr. Michael Rosen, who is chair of the Department of Biophysics at the University of Texas Southwestern Medical Center and an investigator of the Howard Hughes Medical Center. Dr. Rosen is a chemist and a chemical engineer and got his PhD from Harvard University in 1993. He first had a lab at the Memorial Sloan Kettering Cancer Center in New York and then moved to Texas in 2001. Dr. Rosen seeks to understand the formation, regulation, and functions of enigmatic cellular compartments termed biomolecular condensates, which principles stands from the liquid fluid transitions, and which explain a lot of the unanswered or mysteries at the cellular organization and physiology that we have faced for years. I hope you enjoy his talk, and Michael, thank you so much for joining us tonight. The word is yours.
Great. 1 second to share my screen here. Okay. So I want to begin by thanking the organizers of the meeting and Hernandez in particular for the opportunity to spend some time with everybody here and explain some of the work that's been going on in my lab and in general in this exciting field of biological phase separation for roughly the past decade. So I'm going to begin with an assertion, and that is fundamentally life in all of its different forms, from bacteria to plants to fish to mammals to us, is fundamentally chemistry. We are all composed of essentially bags of chemicals, small chemicals shown here at the top, large chemicals, proteins, nucleic acids shown here in ribbon diagrams, but fundamentally we are all basically just chemicals. Now, perhaps a little bit something else, but that's the subject of a different talk for a different day. Now, chemistry is really chemicals plus chemical reactions. And so, for example, I'm showing here a series of reactions that act on the chemical, the molecule shown here on the left. And the bottom line is that chemical reactions change one chemical into another. And so you can see that these reactions here essentially shorten the tail here, the orange tail, by one subunit or two subunits, converting the molecule on the left to the molecule on the right. Now, this is what a simple set of chemical reactions looks like. The problem in a cell is that these chemical reactions occur with a ton of other chemicals and a ton of other chemical reactions. And again, there are many different kinds of small molecules in cells of the body. There are also many, many different kinds of proteins in the cells of the body. And that complexity then creates a problem. And that is, how come you don't get chaos from this? You can imagine lots of ways that you could get molecules that would interfere with each other. Toxic chemicals could be produced by one set of molecules that poison the activities of other set of molecules. How come life can start with chemistries and chemical reactions and produce something that is not chaotic? And a big part of this, the answer to that question, is that this is not the way cells are composed. In fact, cells are very highly compartmentalized. And this compartmentalization goes a very long way to preventing this chaotic mixing of all of the molecules. And so I've sort of indicated here that we have a compartment on the right that has certain proteins and certain small molecules. And we have these other 3 compartments that contain different proteins and different small chemicals. Now, the question is, how is this compartmentalization achieved, which is so central to life? Now, I'm sure that almost everybody in the audience, if not everyone in the audience, is familiar with the idea that eukaryotic cells compartmentalize through creating physical barriers, through using membranes to make different kinds of subcompartments that are sealed off from one another. And those membrane barriers prevent movements of molecules from the inside of a subcompartment to the outside. And in doing so, they create distinct compositions. Different sets of chemicals will be in the nucleus from the cytoplasm, different from the endoplasmic reticulum or the Golgi apparatus. And one can think of this in sort of very simplistic terms as sort of balloons within balloons, that we have a membrane that surrounds the outside of the cell, that prevents diffusion of molecules from the outside air into the balloon. But then within that main balloon, there's also all of these other balloons that have different membranes that, again, seal them off, enable them to be distinct from one another. Now, these kind of membrane-bound compartments have a number of properties that allow them to function in cells. One is that there is actually communication from the inside to the outside, but that has to be achieved through very specialized pores that are necessary for molecules to pass from the outside to the inside or vice versa. The membrane itself is impermeable to most biological molecules. In addition, the compartments generally remain separate, but there are occasions where certain kinds of compartments need to fuse with each other. And that fusion then requires very specialized machinery, such as the protein machineries that I'm indicating here. Further, these compartments tend to have a long lifetime in cells. They exist for days to hours, or hours to days to the lifetime of an organism, some of them. Now, in addition to these compartments that I suspect people have read about in textbooks, it turns out that there's another class of compartments that have been known for decades and decades. Literally, since the images I'm showing here, one of them is from 1898, another one is from the famous anatomist Ramon Cajal from 1910. These are compartments that form without membranes. And again, these very early images of them, taken from some of the earliest microscopy that was done, showed that they are distinct compartments that have distinct compositions from the rest of the cell. These images here show what we've come to call the nucleolus. These foci here, the A foci labeled here, have come to be called Cajal bodies, again, after Ramon Cajal, the one who discovered them. Again, we can see them because they have different compositions. Track forward now to this image was taken from 2017. Turns out that in addition to those 2 very early structures, there are many, many, many now compartments that form in eukaryotic cells in the absence of a surrounding membrane. And these are collectively called biomolecular condensates. Biomolecular in that they are composed of biological molecules, and condensate in that they concentrate or condense proteins and other molecules without a membrane barrier. And I've tried to indicate here in this image, these structures are really found throughout essentially all of biology as far as we know at this point. Everything from signal transduction at membranes to metabolism of RNA to control of gene expression. Kind of any process that you want to look at, turns out it seems to go through a biomolecular condensate. Importantly, and I'll come back to this at the very end, more and more we understand that these compartments, these condensates, are defective in many human diseases. And so not only is it important for basic biology, but they're also important in understanding human health and eventually, hopefully, targeting them for treatment of diseases. Now, in contrast to the membrane-bound compartments, condensates, biomolecular condensates, really have quite different properties. I told you that membrane-bound compartments need specialized pores for molecules to get through them. That's not true for condensates. Condensates quite readily exchange their components with the surrounding cytoplasm or nucleoplasm. And one way you can see this is with these photobleaching experiments, where one bleaches out the fluorescent components of a condensate and then watches them recover over time. So there's the bleach, and then the molecules recover. They diffuse essentially back into the compartment. With the absence of any kind of boundary. I told you that there's complex machinery that's required for fusion of membrane-bound compartments. Not so for condensates. Condensates very readily drip and fuse with one another, and one can sort of watch this movie. If you just look at certain regions of it, you can see condensates that will bang into another one and fuse quite readily, and you can see they have this dripping behavior that is very much like liquids. I told you that membrane-bound compartments have long lifetimes in cells. Again, not so with condensates. Condensates can form and dissolve very rapidly. And so this is a movie showing a condensate called a stress granule, that within minutes of stressing those cells, those condensates form and coalesce with each other. The stress is removed, and again, within minutes, the condensates go away. So very different behaviors of these biomolecular condensates than the canonical textbook membrane-bound compartments. And if you think through these behaviors that I've told you, they suggest quite strongly that condensates behave as liquid droplets inside cells. If you think about the way liquids form, molecules go in and out of a liquid phase quite rapidly. The liquids will fuse and drip around compartments and can form and dissolve or form and evaporate very quickly. So the question then becomes, how do you make liquid droplets inside of cells? And one potential answer, which would seemingly provide a solution to a paradox. How do you push a certain class of molecules to one place in a liquid like a cell? One potential example is the separation of oil and water, or in this case, what I'm showing here is oil, which is the sort of yellow-green liquid, separating from balsamic vinegar, which is this brownish aqueous compartment. So everybody knows you mix oil and vinegar and you get two distinct coexisting compartments. The compositions of the compartments are clearly different. Oil has, if nothing else, has sort of the green colored molecule here, and the water, the aqueous phase, has the brown colored molecule. We all know if you mix oil and water and mix oil and water and watch it over time, if two of the water droplets here, the vinegar droplets, encounter each other, they fuse to make a larger droplet. Something that you may or may not be aware of is even when you see separate oil and water phase, the oil actually has small amounts of water in it, and the water always has small amounts of oil in it. And in fact, the oil and the water are moving back and forth between the two phases very rapidly all the time, even though the phases remain separate compartments. And the other key thing is that compartments form quite suddenly with a change in conditions. So again, something you may or may not be aware of, but if you start with a bottle of oil and you add a really, really, really small amount of water to it, that water actually disperses in the oil. A little bit of water is soluble in oil. You add a little bit more, still is soluble, but if you go over a very sharp threshold, now the water is no longer soluble in oil, and you get separate water droplets floating around in the oil phase. And I'll come back to that issue of how one sees the development of a compartment in just a little bit. So this process, the separation of oil from water, is called liquid-liquid phase separation in the parlance of physical chemistry, and particularly polymer chemistry. And so if oil and water can phase separate from each other, maybe proteins and certain proteins can separate from other certain proteins. And so it turns out that that's the case. There are certain kinds of molecules that either on their own or when mixed together will basically form a separate phase. And what I'm going to argue today is that this is the way that these, at least some of these biomolecular condensates form. Not all condensates form through this mechanism. It's important to note that, but many of them seem to do so. Now, something that has always fascinated me since we began working on phase separation about a dozen years ago now, is that phase separation is not something that is specific to cells. It's not specific to oil and water. Phase separation is something that is all around us in the world. And the concept of phase separation that emerged from the world of physics really is a general organizing principle for the universe that we live in. So I'll just give you a couple of examples, that things that seem completely different, but are all phase separation. So ice cubes floating in water. Ice occurs through a phase separation, in this case a liquid-solid phase separation, from water. Turns out flocks of birds can be considered as phase separation. So this is one of these giant flocks of birds that we sometimes get up here in Texas, and you can watch them float around in the sky. And you have concentrated birds in one region and a few scattered birds floating around them. Mathematically, you can describe that as a phase transition. Traffic flowing down the road and forming a traffic jam can also be thought of as a phase transition. So traffic can uniformly go down the road, go, 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 and then you get one bad actor who decides to change lanes, and all of a sudden the cars clump up into a traffic jam. There are other cars that are on either side that continue moving, but there is a separate phase of cars that are moving more slowly than the surrounding cars. Now, importantly, I'm not making a sort of a trivial point, like, oh, all these things look like a clump of things and then diffuse things around. No, these really are described by the same math, or very similar mathematics. They really, in the eyes of a physicist, are basically the same thing. And they show, because the same math describes them, it's the same basic behaviors that they have. They form coexisting compartments, liquid water, ice water, whoops. The flock of birds and the few birds around them, the traffic jam, and the cars around. They show sudden sharp formation with the changing conditions. The one you're most familiar with, I'm sure, is ice and water. Above 0 degrees centigrade, you have water. Below that, you have ice. When you're right at the middle, the two can coexist, as I'm showing here. Also, molecules move between the different compartments. One bird can separate from the flock and move out, and then a different bird can come back in. And so again, this idea of phase separation, the concept of phase separation, is something that surrounds all of us in the world. And phase separation of proteins inside cells is just one other instance of this very general process. So a key concept that has emerged in this field is that the ability to phase separate hinges on at least in a cell, hinges on the presence of multiple binding elements in the key molecules. So you'll notice that the blue molecule here has 4 different binding domains, and that the tan-colored molecule also has 4 different binding domains, and those can interact in one-to-one fashion. We call molecules with multiple adhesive elements or domains, we call that multivalency. And that multivalency is the key driver of biological phase separation. Now, it turns out that there are, that multivalency-driven phase separation has been studied for many, many years, literally since the 1930s and 1940s, in polymer chemistry and polymer physics. And all of the concepts that arose from those areas of science are now being shown to hold very strongly also for biological molecules and phase separation inside cells. Now, there are lots of different flavors of this. There are lots of ways you can get molecules to stick together through multiple interactions. What I've implied in my previous slide is proteins composed of multiple repetitive domains, when they interact with each other, can undergo phase separation. Those of you who may be familiar with this field will know that there's been a lot of work put into understanding how disordered proteins or disordered regions of proteins can phase separate. It's basically the same concept. A disordered protein will have multiple weakly adhesive elements that can self-interact or interact with other molecules to drive phase separation. RNA and DNA will phase separate through repetitive base pairing interactions. And even as something as large as a synaptic vesicle, which is obviously way bigger than any of the molecules here, if you coat synaptic vesicles with weakly adhesive molecules, when you mix the vesicles together, they will phase separate. And now you get a fluid that's formed not by molecules as the individual units, but by vesicles, with vesicles as the individual units that compose the fluid-like compartment. So, just to summarize what I've told you in the first bit here, biomolecular condensates are cellular compartments that form without a surrounding membrane, and they serve to then organize chemistry because they have different compositions than the surroundings do. Some condensates can form by phase separation, and critically, phase separation is one of these very general ideas and very general processes that even among completely unrelated things can cast them through a common mechanistic framework. And finally, biological molecules phase separate because they have multiple binding elements through this multivalency. So, if we start with multivalency-driven phase separation, there are a whole host of new questions that one has to ask. And I'm going to spend the next part of my talk walking you through some of these. So first, how can phase separation be controlled in cells? Nothing in biology works unless the cell can control it. And second, what does phase separation do in the cell? I began all this by saying we are chemistry, and chemistry needs to be compartmentalized to be able to function. So how is the chemistry affected by this compartmentalization? How does this work to give new functionality? So I'll explain now. I'm going to walk you through fairly systematically, I hope, through some experiments that we've done in my lab to try to answer these questions. And I'll start by saying that when we began this work, we started thinking about, well, what are the multivalent proteins that are out there in biology? And what we quickly realized is that natural multivalent proteins are really complicated. They can contain multiple domains of different kinds, a blue domain, a red domain, a green domain. Even within domains of a similar kind, there can be sequence variations, so there's a blue circle or a blue oval. You can have linkers between them of different length. You can have linkers of different degrees of flexibility. This is sort of a more flexible linker, and this is a more rigid linker. And so starting with natural multivalent proteins is often very complicated. So one of the things that we have done for many years in my lab, in order to really understand these key properties that control phase separation, is we've worked with engineered proteins, man-made proteins. So here's the basic idea. We can start with two proteins, real proteins that bind each other but are very, very simple. So we can take what's called a Src homology 3 or an SH3 domain, and we can bind it, and which is known to interact with its ligand, a proline-rich motif or a PRM motif. Those will form a complex. What we can do is we can use molecular biology, tools of molecular biology, to string multiple copies of these together. So we can take 5 SH3 domains and we can make a single protein that has 5 together, and a single PRM, or a single molecule that has 5 PRMs together. And sure enough, we found that when we made such proteins, I should point out that because the system is very simple, we can learn general principles about what are the features of the molecules that drive phase separation, and then we can go look at the much more complicated natural proteins. So sure enough, when we mix these two proteins and other ones like them together, we can get these beautiful little liquid droplets. So they do phase separate. So again, we can look at molecular features and systematically explore them. So one of the key questions that we like to ask when we find that a molecule can phase separate is, what are the concentrations needed for phase separation? And so we can do the following experiment. We can take the blue protein and the red protein, and we can mix them at different concentrations. And so we can plot concentration of one on one axis, concentration of the other on the other axis, and then we can mix them at a particular point in the 2D array, and we can say, did it phase separate or not? And we can plot that as a color. So in this case, the solution remains homogeneous, no phase separation. We'll give it a blue dot. We can then add little more of the PRM protein, make another point, still clear, still a blue dot. Now, however, if we add a bit more of the PRM protein, remember, phase separation occurs through sharp boundaries. Now we'll see formation of a separate phase. And we can keep this titration going, and we see that we can basically fill in this entire row, seeing phase separation at all of these positions. And again, to come back to this oil and vinegar analogy, I told you that you can start with oil in a bottle, and if you add a little bit of vinegar, the vinegar disperses in the water, and only when you reach a threshold do you form droplets. Well, that's the same that occurs here with proteins. Now, of course, we can make all of these rows again at different concentrations of the SH3 protein, and we can fill in this entire grid. And so again, all of these blue dots means no phase separation. All of the red dots means we do see phase separation. So essentially, this kind of diagram will measure how strongly the system phase separates. What are the concentrations needed of both components to produce this physical separation? So with that assay in hand, one of the key questions that we want to know relating to regulation is, how does the number of binding sites affect phase separation? And to do that, we can do the following experiment. We can make molecules that instead of having 5 domains strung together, we only have 3. We find that when we mix those, this entire diagram is basically blue dots, no phase separation. If, however, we now mix molecules that have 4 domains, now we see that in a lot of the phase space here, we do see phase separation, we do see droplets. And we can do it again with the pentameric proteins. And now we can see on these concentrations, essentially almost all of them produce phase separation. And so what this tells us is that by changing the number of binding sites, we can change the propensity of a system to phase separate. And of course, that immediately begins to think about, to suggest mechanisms of biological regulation. I'll get into that in just a second. So for our non-natural proteins, what I've told you is that these can be used experimentally to simplify our analyses in order to reveal general principles, like the number of binding sites controls the ability to phase separate. And I've told you one of the principles is binding sites, more binding sites, better phase separation. And that this suggests a means of controlling phase separation inside its cells. And now to explain how that control works, I want to now translate this to a real protein system involved in real biology. And so what's shown here is shown here on the right is a system of molecules that we have worked with for a number of years now that produce phase separation and are real molecules involved in real biology. And I'm going to take just a couple of minutes to explain the biology first, and then I'll come back to the molecules and come back to phase separation. And they're involved in biology of the cytoskeleton. So what is the actin cytoskeleton? Well, so the actin cytoskeleton functions inside a cell very much analogous to the way steel girders do in modern skyscrapers. So what's shown here on the right is a picture taken from over 100 years ago of one of the very first skyscrapers being built in New York City. And what you can see is that the center of the building is all of these steel girders, this meshwork of steel girders. And those turn out to provide all of the mechanical support for the entire building. The walls, the white walls that are being built around this structure don't support weight, but they simply seal the structure from the inside, seal the inside away from the outside. Now, in a cell, the cell is very, very highly concentrated in long filaments of the protein actin. So actin filaments fill much of the cell, and they, analogous to the steel girders, provide the mechanical strength of the cell. And the membrane then serves primarily just to seal the cell off from the surrounding environment. Now, in contrast to a skyscraper, a tall building, where everything is static, cells, of course, have to move, and they have to change their shapes in order to move. And so to see that, I'm showing a movie here from Henry Bourne Lab at UCSF. This is a white blood cell moving towards a chemical supplied by this pipette tip over on the left. What you can see is that the cell moves towards the pipette tip, and it does so by expanding its front edge that forms all of these sort of finger-like structures and this big sort of blob that comes out of the front, and it contracts the back end of the cell. It contracts its rear to move the rear forward. Now, for this to occur, the chemistry that's involved in changing the actin cytoskelet has to occur at the right place. There has to be a chemistry that says make more actin filaments here and move the structure this way. There has to be a different chemistry at the back end that says pull the back end of the cell. And so that requires compartmentalization of actin. You've got to compartmentalize it at the front. You've got to compartmentalize it at the back. And so I'm getting to phase separation here, as I hope people can see. So it turns out that these changes in actin are produced by a machine in the cell called the ARP23 complex. And the ARP23 complex then forms actin filaments. And the red circles here are meant to indicate actin. In fact, it forms branched actin filaments that are involved in moving the leading edge of the cell forward. And it turns out that these are activated by this protein called NWAS. And note that the NWAS proteins has a lot of these PRMs, these proline-rich motifs, right? So it suggests that this could be a molecule that could phase separate. And if it did phase separate, that would be a compartment where the ARP23 complex would get activated. And that turns out to be the case. So now we'll get to the full collection of molecules that I want to tell you about. So I told you this NWAS protein here, which is colored red, the thing that activates the ARP23 complex. Turns out in cells, NWAS binds to a protein called NCK, and NCK has a series of SH3 domains that bind to the proline-rich motifs in NWASP. So this bit of the system is analogous to the engineered proteins I was telling you about before, but now it's a natural protein. Critically, NCK also has what's called an SH2 domain, and that SH2 domain binds to phosphate groups that have been added to tyrosines on a protein called nephrin. So we got three molecules, nephrin, NCK, and NWASP. And what's important is that all of them are multivalent. There are three phosphates on nephrin, there are three SH3 domains on NCK, and there are roughly 9 PRMs in NWASP. And so this composition suggested to us, remember I told you more binding sites means better phase separation. This suggested that if you could control the number of binding sites in here somehow, you could control whether you get phase separation or not from this system. And the key thing is that phosphate is a group that can be added and subtracted very readily from proteins by kinases or phosphatases, respectively. And so this was a system that seemed to us like it could be regulated by additional removal of the phosphate groups here. Moreover, it turns out that this is also a system that does in fact make bright foci, bright spots in the cell, and those spots produce actin filaments. And these are some cellular data showing bright spots of nephrin. You can see it better blown up here, and then actin filaments, which are colored red here, emerging from those. And we now know that these spots form through phase separation of these multivalent proteins. And in fact, this is a system that we've shown phase separates both in three-dimensional solution. If we mix all the molecules together, we can see these beautiful round liquid droplets. We can also do experiments, and I'm going to tell you more about these, where we can attach nephrin to a membrane. It's called a supported lipid bilayer, a flat membrane on a glass slide. And if we just attach nephrin, it makes a uniform distribution on the slide. But if we add NCK and NWASP, in fact, we see these clustering of these molecules that have very much analogous properties to these liquid droplets. And so we believe that they also form through phase separation. Now, the hypothesis is that if we change the number of phosphate groups here, if we go from 1 to 2 to 3, exactly as I told you about before, we should get better and better and better phase separation, which is to say that the concentrations of NCK and NWASP that are necessary should decrease as a function of the number of phosphate groups. And so to do that, we examined the ability of each of these three systems to phase separate. So first, we used only the monophosphorylated nephrin here, and we mixed NCK and NWASP to it. So we essentially only have 3 binding sites of SH3s binding to 9 binding sites on the NWASP. And under these concentrations here, these guys actually don't phase separate. You have to go to appreciably higher concentration to see the droplets I showed on my previous slide. Contrast, though, if we now add a nephrin with 2 phosphate groups, essentially what's happened is we've jumped up the number of binding sites from 3 to 6. And now we do begin to see phase separation in this regime. And finally, if we go up to 3 binding sites, 3 phosphates, which is the form that is thought to naturally exist in cells, now we see that there's phase separation across virtually all of this two-dimensional plane. And so what that tells us is that addition or loss of these phosphate groups could control the phase separation in cells. And that's done, those of you may know, through kinases and phosphatases. And of course, since these kinases and phosphatases can act rapidly, that could allow rapid changes in cells in response to signals, rapid formation and dissolution of condensates in response to cell signals. So to sum up what I've told you in terms of regulation, I've told you that the number of binding sites determines essentially the strength of phase separation. More binding sites means you phase separate better. Less binding sites, you have to go to higher concentrations. Phase separates more poorly. And so in the case that I told you about, the number of binding sites can be controlled by phosphorylation. The number of these phosphates matters. And so therefore, phase separation can be controlled by phosphorylation and dephosphorylation. And moreover, this then tells us how cells could rapidly create and dissolve biomolecular condensates, these compartments, in response to various cell signals. So question is, what does this mean to chemistry? Right? I cast all of this in terms of chemistry. And again, I'll remind you that the chemistry that is driven by NWASP is the activation of the ARP2/3 complex to make actin filament networks. And again, to remind you that we think that this could occur inside condensates formed by nephrin and NCK and NWASP. So how do we test this? Well, we can come back to this lipid bilayer sitting on a glass slide. So what's shown here in blue is lipids. What's shown here in gray is the lipid bilayer. And on top of that is a condensate formed by nephrin, NCK, and NWASP. And what's labeled here with the fluorescent protein is NCK. So it's blue here in the cartoon on the left, and it's blue in this actual experimental image on the right. We can then add on top of this, in the same solution, of course, the ARP2/3 complex and actin. And we can add actin in a way that we can see it in fluorescence microscopy. And so the question is, what happens when we do this? Where does actin form when we add them to this system? And so that's the movie that I'm going to show you. So the movie starts with just the condensates formed, and you can see all these puncta on the surface. And then we're going to add actin in the ARP23 complex, and you'll see what happens. So, boom. So we get these beautiful clusters of actin filaments that what you can see, I hope, if I go back here a little bit, is that the cluster, the actin filaments, essentially always start on the condensate. And as time goes by, those grow out and the condensate gets bigger and more of the condensates basically light up with and begin producing new actin filaments. And so what this tells us is the actin filaments then initiate on the condensates and then grow beyond it. What I don't have time to show you data for is that the filament initiation is much faster inside the condensates than in the surrounding membrane. That's why you only see the condensates, that's why you only see the filaments start from inside the condensate. And so just to say what this means, therefore, condensates control where the filaments form and how fast they are produced. And so that tells us how the chemistry is affected by condensation. So to sum up this vignette, phase-separated condensates can recruit activity-bearing components. In this case, we can make condensates, and they recruit actin and the ARP23 complex to make new filaments. And the condensates then spatially localize the chemistry. I've shown you in vitro, but they can also do that in cells. And moreover, the chemical reactions can be accelerated within condensates, which takes us back to the very beginning of how do you deal with the complexity of the cell? Well, you make these compartments, in this case condensates, and the chemistry is then different in the condensates than in the surroundings. So in my last just couple of minutes here, I don't want to keep people too long. I want to come back to one of these ideas that I raised at the beginning, and that is condensates and disease. Because one of the things that a number of labs have shown at this point is that there are a growing number of diseases that result from aberrant formation of condensates. Now, one kind of aberrant condensate can form when there's defective fusing of different genes. So genes such as the BRD4 gene here and the NUT gene here that normally are independent, they're on independent chromosomes. You can get genetic abnormalities that cause these to fuse and make a single gene and a single fusion protein. And in this particular case, what happens is the fusion protein forms a new condensate, an unnatural condensate that doesn't normally exist in cells. And it turns out that this abnormal condensate turns on an incorrect collection of genes. The BRD4 protein is normally a gene regulator, turns on some genes, but when it gets fused to the NUT gene, you make it the NUT fusion protein, a whole bunch of genes that weren't supposed to go on get turned on. And it turns out that many of these genes are involved in driving cancer. And so there are many cancers now that arise from fusion of genes that seem to form aberrant condensates in cells. So if you think about this in terms of therapeutic strategies, a drug that dissolves these condensates could potentially block the actions of this incorrect fusion protein and thus be a way of treating cancer. There's another kind of disease that results from a different kind of perturbation of condensates, and that is neurodegenerative diseases. So in this case, there are many different proteins, among them this FUS protein, FUS, that normally in cells forms very nice liquid-like round condensates. There are many, many mutations now, I'm only showing a handful here of FUS, that give rise to neurodegenerative diseases. Turns out that at least some of those form structures that are no longer liquid-like, but rather these fibrous solid-like things. And somehow those lead to death of neurons and neurodegeneration. For example, one of the most important diseases in this regard is ALS, or amyotrophic lateral sclerosis, also called Lou Gehrig's disease, this terrible, terrible neurodegenerative disease that strikes actually a reasonable number of people, often in their 50s and 60s, and results in death in only a few years. In this case, one can think of therapeutic strategies not so much to get rid of the condensates, but revert an aberrant condensate, this fibrous condensate, back into a liquid condensate. And perhaps that would prevent cells from dying, and perhaps that would allow brains to function and not degenerate inappropriately. So let me just sum up here everything that I've told you. I've told you that this key concept, that multivalency-driven phase separation, can organize cellular chemistry by producing biomolecular condensates. And this tells us a lot about what condensates do. It tells us about the physical nature of condensates, that basically how do condensates form? Through phase separation of multivalent proteins that can then produce these dynamic compartments. The notion of multivalency tells us how these can be regulated, because changes in the number of binding sites, for example, through phosphorylation, will change the propensity to phase separate. I told you a bit how condensation affects biochemical functions, and that certain chemical reactions can be enhanced within condensates, and also then spatially localized in the cell to the place that the condensate is localized to. Finally, at the very end here, I told you, I think, some important medical implications that arise from all of this. The notion of condensates has provided a new understanding of diseases, and also has suggested potential new treatments for those diseases. And I know there are a number of biotech companies. In full disclosure, I've been co-founder of one of those biotech companies called Phase Medicines, that are hoping to target condensates to treat some of these really terrible diseases, cancer and also neurodegeneration, as well as various other diseases. So I just want to wrap up by thanking all of the people who played such important roles in conducting this experiment in my lab. Just to point out a couple of names, I won't walk you through everybody. Sadeep Banjade, Salman Banani, and Wei-Chin Cheng were three graduate students in lab who really launched this whole project in the group starting about a dozen years ago. And I owe all of them a real debt of thanks for their insight and their creativity in moving us in this new direction. So I'll finish with that. Again, thanks to people in my lab. Thank you again to the organizers for giving me the opportunity to speak here this afternoon. And finally, thank all of you for spending your evening here listening to me and learning, hopefully, a little bit about biological phase separation. Thank you.
Thank you so much, Michael. It was a wonderful conference. We learned a lot, I'm sure. I'll give the audience some time for posting the questions, and meanwhile, I will ask my question myself.
Sure.
And do those filaments, actin filaments, growing from the condensates, are they polar, or do they show any polarization? I mean, do we have actin monomers getting into the condensate and then into the filaments, or are they polymerizing from the outside?
Yeah, very interesting questions. Let's see. So maybe I'll start in kind of reverse order. So what we know is that actin, so the condensate is about 90% water. So it's about 100 mg per mil protein, so it's very highly concentrated protein. But if you think about it in terms of water, the condensate is still 90% water. So there's a lot of space in it. And so actin can diffuse in quite readily. The ART2/3 complex can diffuse in quite readily. And those randomly distribute. So they don't have particular orientations as far as we know within the condensate. And so when they start making actin filaments in there, the filaments are moving in all kinds of random directions. Now they get bundled through mechanisms that we really don't understand at this point. And so that's why you can get the images that I showed where you get these very thick looking, those are bundles of actin filaments. But they basically move off in all kinds of different directions. So there isn't directionality because the nucleation starts inside the condensate and because the molecules in the condensate are randomly organized.
Okay. Well, I guess Brazilians are very shy to ask questions. So,
And it's late in the evening for you folks.
okay. It has been a long day. We've been in this conference from 8 in the morning.
Oh,
Okay, there is one coming. Just
somebody said
let
there
it
is a
give some
question.
time.
More than happy to answer.
They are picking down the question from the. Okay, a question from Christopher Kushmerik. You mentioned phase separations of vesicles. Could phase separation explain clustering of synaptic vesicles near active zones in chemical synapses?
Ah, that's a perfect question. The answer is, so that work was done by Pietro Di Camilli's lab at Yale University, and that's exactly the argument that Pietro made in the paper. This was a paper published in Science maybe 2 years ago, or maybe longer ago than that. And that's exactly the point that they were making. The protein they coat the vesicle with is synapsin. And synapsin is believed to play some role in organizing vesicles in the active zone of the synapse. And so that's precisely the argument that they made, that maybe phase separation is the thing that controls the clustering of vesicles at that site in the cell. If you want to send me an email, I'll send you the reference of the paper. I'd be happy to do that. Good question.
Any other question from the audience, Gustavo? Question from Dalton Vassallo. Activation of smooth muscle contraction involves very fast changes.
Let me guess as what the rest of the question could be, and Dalton can come back if
Okay, give it a try.
So the question is, could muscle contraction be something that is controlled by condensates? In principle, yes. I don't know of data suggesting that it is. Activation of smooth muscle contraction involves very fast changes of actin filaments. Increasing the amount near the membrane. Could this be an example of this mechanism? That's completely not the answer the question.
Okay.
Good. That's an interesting thought. That's an interesting thought. I certainly don't know whether that's the case or not, but it could be. It could be possible that one could turn on clustering, because the clustering can occur very, very rapidly, right? In time scales of maybe milliseconds, which is muscle contraction. It might be slower than that, but if so, it could be something that contributes to changes in action there. It's a possibility. Well, I guess I would have to think more about the time scale of muscle contraction and actin dynamics and in contracting muscles, and the time scale of formation of condensates. Whether those are on the same time scale or not, I'm not entirely sure. If they were, this could contribute that.
Very nice. Any further questions, Gustavo? No more questions. Michael, I'd like to thank you again for joining us tonight and for the excellent talk and the excellent conference. I'm so glad you could join us this year. And I hope to bring you to visit Brazil in the near future. So thank you so much.
That would be wonderful, and I'll look forward to seeing you hopefully at an ASCB meeting. Once we can get back to the real world. Thanks again.
Thank you so much.
Appreciate the opportunity. Bye-bye.
Bye-bye. Gustavinho. Gustavo.